Hi class, welcome to chapter 10 lecture. So this is our last lecture in unit 3, and I'm going to divide this lecture into, I think, 2, maybe even 3 portions. But that's it until unit 4. So if you think about what we've learned so far in this unit, in chapters 8 and 9, we've talked about how scientists develop this idea that traits are inherited in a specific pattern. And we call this the chromosomal theory of inheritance, this idea that chromosomes contain genes, and these genes are kind of discrete units of inheritance, and they can be shuffled during meiosis and fertilization to create genetic variation in offspring. And here we have a couple of really good examples of that. I think I'm cutting this off just a little bit. Let me move the, how can I do this? Let me move this just a bit. Down. There we go. All right. So hopefully that's a little bit better. You can see most of the screen. For some reason, the video formatter that I use it tends to cut things off a little bit. And then sometimes it makes my little personal screen here really little, and other times it omits it completely. I'm not totally sure. I'm figuring all of this online stuff out along with you guys too. So let's talk a little bit. Let's do a little review. This is a nice slide because it talks about there's a lot of questions that I can ask you, and I really want you to push yourself to answer these questions. Pause the video, see if you can look up the answers, because these are the kinds of questions you're going to see on the exam. And I want to push you to see if you can remember what these things are called or what terms we might be using and so on. So here we have a female fly. We have two flies, a female fly and a male fly. And you can see that there's two obvious physical differences in these flies, in the coloration of each fly and the wing type of each fly. So let's just say that the B allele stands for, is the allele that determines the color of the fly, where the G allele determines the wing shape. So we would consider the color of the body and the wing shape characters, and then whether this fly is gray in color or black in color, we would call a trait. And so normal wingspan or normal wing shape versus vestigial wing shape would be considered a trait because it's a variation within a character. So we can see that this female, would you consider this female for both, actually it's the same for both, for her color, her coloration, and her wing type, would you consider her homozygous dominant, heterozygous, or homozygous recessive? And ask yourself the same question for the male fly. So hopefully you can see that the genotype for this, so her phenotype is this gray color. Her genotype is capital B, lowercase b. And so remember, when you have two different alleles, that would be called heterozygous. So this female is heterozygous for, I don't call it skin color, but body color, I guess, because insects don't have skin. But I guess we'll call it body color, right? So she's heterozygous for body color. And remember, in a heterozygous individual, the dominant trait is the one that will appear. And in this case, the dominant trait, therefore, must be this gray color. For wing shape, she's also heterozygous. She's capital G, lowercase g, and therefore we know that the normal wing shape would be the dominant trait, because that's the one that appears in a heterozygous individual. Now, on the other hand, we have a male fly that is going to be homozygous recessive for both of these traits. He's lowercase b for body color, and lowercase g for wing shape as well. So here we see that because he's homozygous recessive, the recessive phenotype is going to show up in this particular individual because he's homozygous for both of those traits. So the only time a recessive trait or a recessive allele is shown in the phenotype is if they're homozygous for that particular trait. Good. So now let's talk about the chromosomes themselves. So how many chromosomes do you see here? Let me see if I can circle it. I got one of these fancy little pens that kind of writes on the screen, so I'm hoping this works. Hey, there we go. So how many chromosomes do you see there? And here, right? Hopefully you can tell right away that we see two chromosomes, because remember, if you're ever if you're ever, and I still get confused about this to this day, whenever you're confused about counting chromosomes, count the centromeres. Remember, the centromere is that little just kind of pinched in portion of the chromosome. So you can see the centromeres are here, and you can see two centromeres and therefore 2 chromosomes here, 2 centromeres and therefore 2 chromosomes here. And that makes sense because remember, we're going to get a chromosome from our mom. So let's just imagine that this top chromosome we got from mom and this bottom chromosome we got from dad, and the same for the male fly over here, one from mom, one from dad. So when we look at these 2 chromosomes, what's the term that you would use for these 2 chromosomes? What would be the term that you would use? So hopefully, again, you recognize that these would be considered homologous chromosomes because homologous chromosomes have the same alleles in the same locations. And again, we have 2 copies. They're not really copies because they're not exact replicas of one another. They have different genes, like very clearly there's different genes in each of these chromosomes, but we get 1 chromosome from mom and 1 chromosome from dad. So we say that we have 23 pairs of chromosomes in a human, and therefore we have 46 total chromosomes. So we consider these homologous chromosomes. Same over here, right? These are homologous chromosomes. Now, let's look at what happens in gamete formation. Remember, in order for these 2 flies to create offspring, they have to create sex cells, also known as gametes. And these gametes are formed through the process of meiosis. And meiosis, the whole point of meiosis really is to create genetic variation in these sex cells so that when let's just say that these 2 flies mate, that their offspring will have lots of genetic variation in those offspring. So let's ask ourselves again, test yourself again. How many chromosomes do you see there? Remember, count the centromeres. 1 centromere here, 1 centromere here. There are 2 homologous duplicated chromosomes here, right? So we would consider, so these are homologous chromosomes, whereas these would be also homologous chromosomes because they contain the same allele, same genes in the same location, at the same loci, but they're duplicated. We can see that we now have sister chromatids on each of these chromosomes. So these 2 duplicated chromosomes together, what is the correct term? We see it over here as well, right? We see 2 duplicated homologous chromosomes. What would we consider, what's the correct term to use here? These are called tetrads, and tetrads form in, what phase of meiosis? You should know this, right? What phase of meiosis do tetrads form? They form in prophase 1. Yeah, tetrad formation occurs in prophase 1 of meiosis. So this next little process here is yet another really good test question that I could ask. What happened in that third circle down here? What mechanism or what phenomenon just happened? You can see that these sister chromatids now look very different. These duplicated chromosomes look different. What occurred to make that happen? And so hopefully you can see that they switched pieces of their chromosome. Remember when tetrads get really, really close together, they can kind of just spontaneously switch their chromosomes, and that's called crossing over. So we can see crossing over. And remember, the value of crossing over is to create more genetic variation in those gametes. And so when in this case, this female fly produces eggs, now all 4 eggs are going to be different. And whereas if crossing over didn't occur, she would only have 2 different possible combinations of, excuse me, 2 different combinations of gametes. Now she has 4. So she just doubled the genetic variation in her offspring by doing, by having crossing over occur in gamete formation. This also occurs in prophase 1. This occurs in the first part of a very early stage of meiosis. So Because the father is homozygous for both of these traits, right? There is no genetic variation in these two alleles. The father really can only produce one type of sperm based on these two alleles. Because the mom is heterozygous for both of these traits and crossing over occurred, the mom, the female parent, has 4 possibilities in her gametes. So the last thing that I want you to do is determine what the genotype of these 5 gametes would be. So pause the video and see, and again, all the information is on the screen, so you should just be able to follow the genotypes here to determine what the genotype of each of these gametes might be. So pause the video and see if you agree with what I have. So you can see that this is going to lead again to 4 variations in 4 variations in her in the female parent's gametes, and this occurs. So the first, the tetrads, right? The two chromosomes are going to pull apart in anaphase 1, and then the sister chromatids are going to pull apart in anaphase 2. So in anaphase 2, we're going to have a capital B, capital G. That's going to go here. We have a capital B lowercase g, which is going to go right here. We have a lowercase b capital G right there. And then our last possibility is lowercase b lowercase g, which would go there. So I hope the pen is helping. I'm a little pen happy. I'm excited to play with my new toy. So I hope this is helping a little bit. So again, because of independent assortment and crossing over, we're going to get 4 different kinds of, right? I'm pointing at the screen now. We're going to get 4 different kinds of gametes that the female parent can produce. Whereas because the father is homozygous recessive, he can only produce one combination. There's no other, oh wait, this should not, why is this? That should not be a capital B, that should be a lowercase b. That's a typo. I bet it did that automatic caps there. So the only possible combination that the father can produce, no matter how he goes through, how his sex cells go through meiosis, is lowercase b, lowercase g. And then of course, when this sperm fertilizes one of these eggs, that is called fertilization, a zygote would form, and that zygote, we could do a Punnett square to determine what the genotype and therefore phenotype of that offspring could be. So the question is, what we're going to answer in this chapter and in this lecture is what is B and G? Like, what does that even mean? Like, how does that lead to the physical appearance of a trait? And what is a chromosome exactly? Well, a chromosome is made up of 2 of the 4 major macromolecules. Remember that we have 4 different types of macromolecules. We talked about these way in maybe even like in the second week of the semester. We have, actually see if you can name them. Think about what the 4 major types of macromolecules are. Remember, we have lipids, we have carbohydrates, proteins, and nucleic acids. And and chromosomes, these chromosomes that we inherit from our parents, are made up of 2 of them. They're made up of DNA, which is a type of nucleic acid, and protein. All of the inherited material, so the alleles and the genes that determine our traits, are found in the DNA. And the proteins really act like a scaffold that supports the DNA and how it sits in our nucleus. So we have 2 major kind of pieces of our chromosomes, DNA and proteins. All right, so we're going to we'll talk about proteins a little bit later in the lecture, but for now, let's talk about the important molecule, the most important molecule in terms of our chromosomes and inheritance, and that's, of course, the DNA. And we want to know the structure of DNA because, again, we ultimately want to understand how DNA is able to demonstrate inheritance. We know that DNA has to somehow code for a black-bodied fly versus a gray-bodied fly. We know that there's somewhere in the DNA is a code that will tell that this particular offspring to have normal wings and this particular offspring to have vestigial wings. So we want to drill all the way down to the actual structure of DNA and try to figure out if we can see how that code is formed. And we can, right? That was the big advance of the 20th century in the elucidation of the structure of DNA. And we now understand exactly how DNA works. And it's really pretty cool. So I'm sure you've seen this picture of DNA. It's a very famous kind of model of what DNA looks like. A DNA is considered a double helix, and helix is, again, another one of those annoying science terms that really just mean a twist or a spiral. So a helix just means something that's been twisted on itself or is spiral shaped. And it's considered a double helix because there's two strands that make up this this twisted structure. So imagine, if you will, a ladder. Everyone, think about what a ladder looks like. And if you were to take that ladder and somehow twist it on itself, that's kind of what DNA that's what DNA looks like. So if we were to look at one of the strands of DNA, we have two different ways that we can look at it. Here's a simplified view of each strand, where we see all of our little base, our nucleotides, A's, C's, T's, and G's. But we can look at it in a little bit more detail. So again, these are two representations. Here's a simplified representation. Here's a little bit more complex of a representation. And I want to focus on this one for now. So in DNA, we have two regions, if you will, of DNA when we look at a single strand of this double helix structure. So the blue shaded region, we're going to call a sugar phosphate backbone. So again, if we're thinking of a ladder, it's the sides of a ladder. So the vertical part of the ladder. And in DNA, it's the same. It's always the same for everyone. In other words, your sugar phosphate backbone and my sugar phosphate backbone look exactly alike. In fact, the sugar phosphate backbone for all organisms, for all life forms, looks the same. Again, I think that's pretty cool. Amoeba, bacteria, lizard, duck, flamingo, all of us, we have the same sugar phosphate backbone to our DNA. And we call it a sugar phosphate backbone because it's made up of, you guessed it, sugar and phosphate. So this little pentagon shaped molecule here is actually a sugar. And the circle, the yellow circle, is a phosphate. And I'll go into a little bit more detail on what that means here in just a second. But notice that it alternates. We have a sugar and then a phosphate, a sugar and then a phosphate, a sugar, phosphate, and so on. So this composes our backbone, and it's always going to be the same. We always see this repeating pattern of a sugar, phosphate, and so on. Now, the other portion of DNA is what makes us unique. So these nucleotides, the nitrogenous bases that are sticking out of the sugar, notice that it's connected to the sugar. Each time we have a sugar, we have this nitrogenous base that's kind of sticking out the side of it. And this is what gives us our variation. Right? If DNA is different, right? We all know that all of our DNA is unique. Your DNA is kind of like your finger. It's actually better than your fingerprint. DNA is way more accurate than fingerprint analysis, believe it or not. And DNA is unique to each person. The only exception to this is in the case of identical twins. DNA, the sequence of how these A's and C's and T's and G's are put together is unique to each person. And here is where we get our variability. And so this part of the DNA sequence is called a nitrogenous base. So when we think back to, again, that second week when we talked about nucleic acids and how they are a polymer, the monomer of nucleic acids are called a nucleotide. So you can see pretty quickly here that these repeating units of nucleotides are made up of these three pieces: the sugar, the phosphate, and that nitrogenous base. So in this case, the nitrogenous base is thymine, but it can be any combination, right? It can be any of these: A's, G's, C's, or T's. So let's look at this sugar in a little bit more detail. Who knew that our DNA was made of sugar? I think that's kind of a weird concept to think about. But the sugar that the sugar that's used in our DNA is a 5-carbon sugar called ribose. And so the form of ribose that is used in DNA is called deoxyribose. And we know that this is a carbohydrate, a sugar. Remember, sugars are types of carbohydrates because it ends in OSE. Ribose is a type of sugar just like glucose, sucrose, fructose, lactose, and so on, right? That OSE is going to tell us that this is a sugar. And this particular sugar has 5 carbons in it. Let's count them. 1 carbon here, 2, 3, 4, and 5. And something that I want to point out here that will become important later is that our nitrogenous base is connected to this first carbon. We're going to call this carbon number 1. And as we work around carbon number 2, 3, 4, and 5, our phosphate group is always going to be connected to that 5th carbon, that carbon number 5 over here. So this is our nucleotide, the 3 units, the 3 subunits of our nucleotide. We always have a phosphate group and a sugar that makes up our backbone. And then we have a nitrogenous base, one of 4, A, G, C, and T, that make up kind of the pieces of the ladder that you step on, right? The places, the part that you actually use to climb up is made up of our nitrogenous bases. All right, so let's look at these nitrogenous bases in a little bit more detail. Sorry, I just want to adjust this just a little bit. All right, okay. Our nitrogenous bases fall into 2 categories. We're going to call the thymines and cytosines. Notice that they both have a single ring structure. We're going to call them pyrimidines. And the adenines and guanines are both made up of 2 ring structures. You can see the double ring structure in both of these, and we call these purines. And so various students come up with a mnemonic to remember this because you get to use your notes for the test. I guess it's not terribly important that you memorize this, but understand that these nitrogenous bases do have different structures, and the structure actually becomes important because it dictates which nitrogenous base can pair up with the other. So let's talk about that. I think that's right in the next slide. And also, I don't think I've talked about the names of these, right? So A stands for adenine, G stands for guanine, cytosine is C, and of course thymine is T. So I think I have a couple quick slides on how we actually discovered the structure of DNA. So up until, goodness, probably like the 30s, maybe the 30s I'd say into the 1930s, scientists still did not know what the point of DNA was. So they could take cells, they could take a lot of these early experiments were done with bacteria or with different types of fungi, and they could separate all the chemicals, they could separate all the macromolecules that made up, let's just say bacteria, for example, and they could see that we have lipids and carbs and proteins and nucleic acids, and they knew what proteins kind of did. They could see that lipids formed the cell membrane and carbohydrates were sugars and they had various functions within the cell, but no one really knew what the point of DNA was, and it was really kind of ignored for a really long time because scientists just didn't know what the point of it was. And up until the 30s, most scientists at that time actually thought that the molecule of inheritance was proteins. And so it wasn't until the 30s, even into the 40s, that there was really strong evidence that DNA was actually the molecule. Nucleic acids, DNA and RNA, were the molecules of inheritance with a couple of important, very important scientific experiments. And so late in the 40s, but they still didn't understand. So there was this big advance and everyone was starting to accept that DNA was the molecule that contained these so-called genes, right? These packets of inheritance. But we still didn't understand, scientists at that time still didn't understand how it actually worked. How did DNA get put together? What was the structure of DNA? And so a huge advance was made in the early 50s by Rosalind Franklin. She was a researcher at, I believe she was at Cambridge in Britain. And she was a scientist she was an X-ray crystallographer. And X-ray crystallography is a method of trying to see what the 3-dimensional shapes of molecules are. So imagine if you're in a room and you can't, everyone's played like, what is it called? Like shadow puppets, right? If you have like a flashlight and you put your hands a certain way and it makes a reflection on the wall wherever you're pointing the flashlight. That's kind of like, it's a very simplified explanation of what X-ray crystallography is. So you basically take a crystallized version of the molecule that you're looking at, and you fire electrons at it, and you want to electrons. And you want to see how those electrons are refracted. In other words, how they bounce off that molecule. And you look at the picture that they make on the wall. So you're not really looking at the molecule itself. You're looking at the picture or the shapes that that molecule makes when you're looking at the reflection. So Rosalind Franklin was looking at DNA, and she wanted to know what the three-dimensional structure was. And she came up with this very, very famous picture. It's called, I think it's called, what is it called? Photo 51, I think. I feel like I should remember this, but because it is such a famous picture. It's like, this picture is literally in every biology book that's ever made, because it was the picture that was the last piece of the puzzle for the scientists that are credited with the discovery of DNA. And of course, that's Watson and Crick. So Watson and Crick saw Rosalind Franklin's picture, the very famous X-ray crystallography that showed that DNA was a double. Believe it or not, this picture basically shows us that DNA is a double helix, and showed that refraction pattern that allowed Francis and Crick to make the realization that DNA was put together in this two-stranded twisted model called, of course, now called the very famous double helix. So they were able to update their DNA model, and of course they won the Nobel Prize in, I think, the early 60s for this discovery. So let's talk a little bit about how this structure, elucidation of the structure, basically opened the floodgates. So if you know, so it's kind of like, how would I say this? Like, imagine if we had a car, like we all knew that cars are able to drive us around and get us from place to place, but we didn't know how cars actually worked. We didn't know what the inner structures were, and therefore we didn't know how to fix it, right? And so if the scientists made this discovery, like, oh, this is how cars are put together, the pistons work like this, and it turns the axle, and the axle turns the wheels, and so on, right? We now know how all these pieces of the puzzle come together, and now we know how to fix it, we know how it works, and we can play with the different components and modify them and so on. And that was, of course, the big advance that brought forward, I should say, the modern era of biology. And we have now kind of moved into the genetic era of biotechnology because of this discovery of the structure. So one of the big rules and the big advances of the elucidation of the structure is that we now know that there's a very simple base pairing method to DNA, and we call this Chargaff's rules, and that's a kind of a rule for base pairing. We can see that because a purine, for example, adenine, will always pair up with a pyrimidine, in this case thymine, we can determine the structure or the entire sequence, I should say sequence. We can determine the base pairing for the entire molecule of DNA. Because we know, again, this is an always, always, always thing, in DNA, adenine will always, always, always pair up with thymine. Guanine will always pair up with cytosine. And because of this very important rule, this Chargaff's rule, if we know the structure of the number of adenines in DNA, then we know that the amount of thymine will be exactly the same. And then we can actually determine the amount of guanine and cytosine as well. So let's see if you can figure this out. If you know that 20% of your base pairs are adenine, so 20%, if you were to sequence your DNA and you realized, or the scientist or the doctor, whoever it is, told you that 20% of your nitrogenous bases are adenine, how much would thymine be? So remember, if 20% is adenine, and adenine and thymine always pair together, you know that thymine has to be the same, right? We have to have 20% be thymine. So here's the question. Can you figure out how much C and G there are? Pause the video if you can't figure it out. Take some time and ask yourself how much of your DNA would be guanine and how much would be cytosine. Well, we know that if 20% is adenine and 20% is thymine, then we know that roughly 40% is going to be due to A's and T's, and therefore 60% has to be due to our guanine and cytosine. And since we know that they have to be equally distributed because they're paired, we know that 30% has to be guanine and 30% has to be cytosine. So knowing one, we started off simply by knowing adenine. Knowing one will be able to tell us the percentages of everything else. This is Chargaff's rule. And this was a very handy rule when scientists were trying to sequence DNA and figure out, basically map where genes are on chromosomes of bacteria and those early models of biotechnology. This slide reminded me of something I forgot to talk about, and that is hydrogen bonds. Let's talk about hydrogen bonds. Oh, here, great. I can talk about it now. I didn't forget. That's just saving it for now. So now that we know that A's always have to pair up with T's and C's always have to pair up with C's always have to pair up with G's, we can kind of get an idea of how these two, remember, because DNA is made up of two strands, how these two strands fit together. So one strand, remember, we always have that, where's my little cursor here? We always have our sugar phosphate backbone. Here's our sugar phosphate, sugar phosphate, sugar phosphate, and so on. Notice that we see it on this side and on this side. And our nitrogenous bases are going to stick out into the interior of the DNA model. And again, we have T pairing up with A, C pairing up with G, G pairing up with C, and so on. And notice that these two strands are held together by a hydrogen bond. And this is one of the coolest things, I think, and it just blew my mind when I kind of really understood this. And because remember that hydrogen bonds are really weak. They're super weak bonds. They're kind of like Velcro, like you can take them apart and put them back together. They're nothing like covalent bonds, right? These, like this phosphate is connected to this sugar molecule by a covalent bond, which is a really strong bond. That bond's not going to come apart. But the two strands of our DNA are held together by basically the weakest kind of bond, a hydrogen bond. And you can see that A's and T's are held together by two. You can see these kind of dotted lines represent the hydrogen bonds, whereas the C's and G's are held together by 3 hydrogen bonds. So a hydrogen bond on its own is super weak, but multiplied by the billions and billions and billions and billions of hydrogen bonds that exist in this strand of DNA, it can become strong enough to hold those two strands of DNA together. And this is why DNA is double-stranded. It's held together by these hydrogen bonds. And what's really kind of cool is because it's held together by hydrogen bonds, it means that those two strands can come apart. And that actually becomes very important for expression of genes, for example, and replication. We need these two strands to be able to come apart, but then also come back together again. And that's exactly what these hydrogen bonds allow us to do, because they're kind of like Velcro. Remember, Velcro, come apart, put together, come apart, and so on. So if you noticed, let me go back to this slide really quick. You probably noticed something that looked kind of weird to you. If you looked at this strand or this side of the DNA, it looked kind of like as I was describing it, sugar phosphate, sugar phosphate. But if you look at the other side, everything kind of looks upside down. Well, that's because it is. DNA strands have a very specific orientation to them, and it turns out that these two strands are in opposite orientation to one another. So let's talk about orientation when we're talking about DNA. And remember that when we talk about a biomolecule, we really can't use like terms left and right or top and bottom, because DNA exists in 3 dimensions, right? It's in our nucleus and it's tumbling around and it's in 3. So as soon as it's left and right, it might turn around and now that's all switched around. So we need to be able to discuss DNA in terms of its directions in a specific way, and we can't use top, bottom, left and right, and so on. So what we're going to do is we're going to describe directions based on its carbon atoms. Remember when we looked at this sugar, here's our ribose sugar, and here's our phosphate group. Remember when we first looked at this, we numbered those carbons, 1, 2, 3, 4, and 5. And that phosphate group is always on that 5' carbon. So the end of DNA that ends in that 5' phosphate is always going to, and so just a little bit of organic chemistry. When we talk about the carbons and that little apostrophe, we call that prime. No, it's just kind of the nomenclature of science. So that number 5 carbon, you could say, is called the 5' end of this particular molecule. So the 5' phosphate will be on one end. And if we go down to the other end of the DNA, we're always going to end with that 3. So here's carbon number 3, or the 3' carbon. We're going to end with that hydroxyl group down here. So in terms of directionality with DNA, we can talk about one end being a 5' end that will always have the phosphate group on it, and the other end being the 3' end that will always have either the sugar on it, or more specifically that hydroxide group on it. So when we talk about DNA, the two strands of DNA are put together. Remember, they're connected by their nitrogenous bases, but they run in an anti-parallel fashion to one another. And so anti-parallel means that if one side of the DNA molecule is the 5' end, the other side will be the 3' end. So they're really, it's kind of like I kind of think of it like a two-way street, right? Two-way street, like streets that are two-way streets, yeah, are parallel, right? The lanes of traffic, hopefully, are, if they're not, they're in big trouble, right? But they're always parallel to one another. In other words, they're never going to cross one another, but they run in opposite directions. So this side of the DNA molecule will run from 5 to 3, whereas this one will run from 5 to 3, but going the other way. So 5'. Okay. All right. So Watson and Crick's model was such a big advance, not only because they solved the puzzle, right? They solved the puzzle that everyone in the world at that time was working on, but because they solved that puzzle, their model for how DNA was put together suggested a model for replication. And that was the thing that really kind of stumped scientists for a long time, is if we have this molecule of inheritance, how does it actually make copies of itself to pass on to either a new cell through mitosis, right? Think about it. Right now, we have skin cells that are dividing on our body. We have red blood cells that are dividing. We have all kinds of cells that are undergoing mitosis. So the question that the scientists were asking is and each one of those cells, right, when we undergo mitosis, each one of those daughter cells gets a new copy, a new chromosome, a new nucleus, new organelles, a new everything, including DNA. So for Watson and Crick's model showed a model of how DNA could replicate itself. And because it was two-stranded, right, our parent molecule has these two strands that are complementary to one another. In other words, if this is an A, we know this has to be a T. If this is a C, this has to be a G. So imagine if you didn't see this side of the DNA molecule. You would know it because you know what the other side is. And so that's called a complementary structure. So if we were to separate these two strands, we could actually make a new strand that would connect with this side, and we could make a new strand that connects with this side. And in fact, that's exactly what happens during DNA replication. The two parent strands separate, so they pull those hydrogen bonds apart, and new nucleotides are added to each of the strands. And so when daughter strands form, you'll see that we now have two perfect copies of this original parent molecule. And these two new copies contain one strand that was from the parent and one entirely new strand. So in this way, we consider DNA replication to be half conserved. In other words, one parent molecule, one parent strand is conserved, and one strand is completely new. And this model of replication is called semi-conservative replication. In other words, half of the original, right, this portion, right, this strand here is here, and this strand here is here. And so this is called semi-conservative. So every time DNA makes a copy of itself, half the material is old and half the material is new. And we get perfect copies pretty much perfect copies every time. So if you recall, DNA replication occurs in the S phase of interphase. So before the cell undergoes, in this case, let's just talk about mitosis and keep it simple. So before the cell enters the mitotic phase, so during S phase of interphase, the DNA has to replicate itself. If DNA replication doesn't occur, the cell can't divide because it doesn't have two copies of DNA. And so the first thing that has to happen is the double helix unwinds itself. And so it starts to truly look like a ladder, right? And can see kind of the rungs of the ladder, right? Here's our sugar phosphate backbone. And here are our nitrogenous bases that make up the middle part of our DNA. And our DNA is going to separate. So you can see the top strand here is in a 3' to 5' direction. And our bottom strand is, of course, in the opposite orientation. And a very special enzyme called DNA polymerase is going to come in and start adding nucleotides to each of these strands. And so we know that this is an enzyme because it ends in ASE, polymerase. If you think about that term polymerase, remember poly means many. And so we're going to make a new strand of many DNA nucleotides together. So we're going to stitch those nucleotides together based on a template of the old, so the blue strand here. And so DNA polymerase can add these nucleotides one at a time and this is very important, in a 5' to 3' direction. So DNA polymerase is a unique polymerase in that, I shouldn't say unique, all enzymes have to work in a certain fashion. They have a certain method of how they work. Like I'm trying to think of an example that we could use. Like something that's only one way. What's something that only works in one way? Goodness, I'm having a hard time coming up with a good analogy for this. Like, I'm stumping myself right now. I feel like I have a good analogy for this, but I can't seem to come up with a good analogy that would relate this to something in real life. I'll come up with something. But this DNA polymerase can only add nucleotides in a certain direction. In other words, it can only add nucleotides to the 3' end of this growing DNA strand. I think I have better detail in the next slide. Next slide? Okay, yeah, it is in the next slide. So I'll come back to this slide. I want to kind of finish this thought on the directionality. So DNA polymerase, because it can only synthesize a new strand in a 5' to 3' direction, the DNA polymerase can follow the replication fork in a continuous way in this top strand. But in the bottom strand, it kind of has to work backwards. And so because it can't go this direction, because then it would be adding the nucleotides in a 3' to 5' direction, and that just doesn't work. So when we talk about replication, so again, I apologize, I'm going to go back one now. The parent DNA molecule, remember, we have 23 pairs of chromosomes. We have an enormous amount of genetic material to replicate prior to entering mitosis. And so if we started replicating our DNA on like chromosome 1, position 1, and then we had to go through all 46 chromosomes, it would take a year to replicate one copy, just to make one copy of our DNA. And so what happens here is we have multiple origins of replication. So these origin of replications, in other words, we have multiple sites along our chromosomes where we're going to have a bubble form and DNA is going to start to replicate. So the strands, the blue strands are our parent strands. They're going to pull apart and our DNA polymerase is going to start making that new genetic material. It's the gray strand. And when those gray strands meet, we can see we now have completed replication, and we now have an original parent strand, the blue strand, and a new daughter strand, that gray strand. So again, this is a little bit of a difficult concept for students to understand, and there's a great Amoeba Sisters video on this specifically about continuous and discontinuous replication. The leading strand, so this top strand, we're going to consider, we call it a leading strand because the DNA can be synthesized continuously. In other words, the DNA polymerase can go right along the replication fork and add those nucleotides one after the next. Now, on this case, it's the bottom strand. The DNA polymerase can't do that. It can't follow in the same direction because remember that the DNA strands are running in opposite directions. So in this case, the DNA polymerase has to work in a discontinuous manner. And they basically make very small pieces of DNA. And then another type of enzyme called DNA ligase comes in and stitches all of these small pieces together. And so this is kind of going into a little bit more detail about what I was talking about. The nucleotides with DNA polymerase, it can only add nucleotides to that 3' end. It can't add a nucleotide up here on the 5' end. So we can only make DNA grow on that 3' end. So we can only add this new nucleotide to this growing strand here, never here. So never up here. So here we can see, here's our original parent strand, and we have our growing strand here, and we can only add this new, in this case a thymine, this new nucleotide, to the 3' end of this particular growing chain. So DNA polymerase can only add nucleotides, and so here we can see it's fully attached, and the next nucleotide is going to come in here and attach at the 3' end of this nucleotide. What nucleotide would be added next? Well, if this is a C, this has to be a G. This has to be guanine, right? The only nucleotide that could come in here is a guanine, and it will be added to the 3' end. All right, so let's practice this. Let's see if we can replicate these two strands. So we have a small stretch of remember our DNA is billions of base pairs long, but let's only do a few of them just for practice here. And let's see if we can replicate the DNA for each of these strands. So we see our two strands, remember they're in opposite orientation to one another. And in order to do this, let's, oh, nice. Okay, we're gonna pull them apart just a little bit, and let's replicate the top strand first. So go ahead and pause the video, see if you can work out not only what the base pairs will be, but what direction should this go in. Just a little bit. Pull this in a little closer. So if this is the 3' end, the new chain. Sorry, this is in red. It's the only ink color PowerPoint will let me use. So the new strand has to be in opposite orientation, and because DNA polymerase can only work in this direction, this is the direction that we will kind of replicate here. So we're going to, DNA polymerase will add to this nucleotide first. So C, the complement to C is always G. T would be A is T, C, G, A is T, A, G, C, C. Look at that. You just replicated your first strand of DNA. So now let's try the bottom strand. So the bottom strand is 5 over here and 3 over here. So that means that we have a 5 to 3 prime direction here. And again, we always replicate, or we always make new DNA in a 5 to 3 prime direction. So now we're going to go G, C, wait, no, G, sorry, G, C, T, A, T, T, C, G, A, T, and C. There you go. You just replicated DNA. Congratulations. All right. Here's another little concept question. When a DNA molecule is replicated to make two daughter DNA molecules, the daughter DNA molecule contains how much of that original parent strand? The correct answer here is B, 50%. So if we have our original parent strand, remember they're going to get separated during replication, and we're going to make a new daughter strand based on that template. So half of each daughter cell is going to contain the parent DNA, and half of it's going to contain new DNA that was added, right? So if we go back to this, right? If we look at each new molecule of DNA that we made, half of it is our old DNA, and half of it is the new nucleotides that we made, that we put on there. We, of course, being DNA polymerase. All right, I think there's one more here. And our last concept question, which of the following is listed correctly in order of increasing size? So you're gonna have to put, we haven't actually directly addressed this yet, but hopefully you recall that, so what we kind of focused on, sorry, I'm like half out of the picture here. What we kind of focused on in this this portion of the lecture were nucleotides, what makes a nucleotide. Remember, there are three pieces: a phosphate, a sugar, and a nitrogenous base. And those nucleotides come together to form a string of DNA. That DNA can code for a gene, and genes, of course, are located in chromosomes. So the correct answer here would be E, elephant, is the correct answer. So I think I'm going to cut it off here. We're already over 50 minutes. This is a long one. And when we start our next lecture, we're going to talk about how DNA is actually made into protein. So spoiler alert, DNA is first made into RNA. We'll talk about what RNA is here. And then we'll talk about what proteins are and how we actually get from DNA to proteins. Stay tuned.